Human nutrition is our main thing today. So, human nutrition. Can you all see my whiteboard?
Yes, sir.
Yes.
Yes,
So
sir.
under human nutrition, let's quickly look at certain things. So I'll start from carbohydrates, yeah. Now, what are carbohydrates? What are they made of? What elements make up carbohydrates? Some of you,
Hydrogen, carbon, and oxygen.
yes. So carbohydrates are made of carbon, hydrogen, and oxygen. Morning. And
Sorry, sir, could you just let me record, please?
yes, we said carbohydrates are made of carbon, hydrogen, and oxygen, CHO. Those are the only elements, no other elements, that make up carbohydrates. Are we together? Karen, you're there?
Yes, we're together. Sorry, sir, could I record the lesson, sir? You need to grant me permission.
Okay, but I was recording it. I thought last time it was affecting my pen, but anyway, let me try. Seemed to affected my pen after I did that. Okay, anyway. Okay, that's fine. So we're saying carbohydrates are made of carbon, hydrogen, and oxygen, CHO. What are the uses of carbohydrates? Is Ella in the lesson? Ella is she? Is that what she said?
Sure.
Yes, sir.
You said the network is bad.
Yes, sir.
Okay, we are recording. So carbon, hydrogen, and oxygen. Those are the three elements. Now, what are the uses of carbohydrates?
Energy provision.
Yes, carbohydrates provide energy. Uses. They provide energy for what? What is the energy used for?
Provide energy.
Yes. For what? Not for the body. Yes, that's a core answer. Energy for what? What are the uses of respiration? Number one, respiration uses energy. What else uses energy? What are the uses of energy? Doesn't active transport make use of energy? Active transport, because it's against concentration gradient, it has to use active transport. What else? Energy. I think some guys will come back. What are the other uses of energy? Energy, respiration, active transport, what else? Movement.
Performing physical tasks.
Okay, so movement in animals is called locomotion, skeletal movement. Locomotion is skeletal movement. Are we together? Movement in search of food. Okay, are we together? What else? Energy is used for what? Transmission of what? Transmission of nerve impulses. Of nerve impulses. Makes use of energy. Clear? Energy is used for cell division. For growth, we call that cell division, mitosis, and meiosis. They use energy. Are we together? Is that making sense?
Yes.
Okay, now what are the three types of carbohydrates? There are many uses of energy, but I'm just giving you the 5 for now. What are the three types of carbohydrates? What are the three types of carbohydrates? Oh, is that new to you then? You look surprised. Three types, there are three types. Carbohydrates are found in three types. Yes. Tambelani, you're raising your hand. Your hand raised. What are the 3 types? Monosaccharide.
Saccharide.
Have you never heard of that? Monosaccharide.
Monosaccharide.
What is a monosaccharide? A monosaccharide, what is it? It's the simplest, it's made of one carbohydrate molecule. Huh? One carbohydrate molecule. Mono means one. One carbohydrate molecule. Clear? By one carbohydrate molecule, I'm simply talking of C6H12O6. Those doing extended, you need to know this. This is the one carbohydrate molecule. C6H12O6. So it means monosaccharides have the formula C6H12O6. Clear? They are soluble or soluble in water. They are all soluble in water. What are the examples of monosaccharides? They are the simplest sugars. So what are the examples? Glucose. Remember when you're writing photosynthesis, you write this. Glucose. What else? Glucose. In fruits, what do you find in fruits? In fruits, what do we find?
The
Yes.
fructose.
Fructose. What else? Fructose.
Galactose.
And galactose. Those are the 3. Glucose, fructose, and galactose. Are we together? And they are mainly found in fruits and honey. Yeah? Main sources for this is fruits and honey. Clear? Now let's look at the next one. Disaccharide. What is a disaccharide? What is a disaccharide? Di means what? You can guess it. 2 what?
Yeah.
Yes. So they consist of two. Yes. 2 carbohydrate molecules. Huh? Or let's say it consists of 2 monosaccharides joined together. So that's the proper. Huh? 2 monosaccharides joined together. Clear? Consists of 2 monosaccharides joined together. Making sense now? They are also all soluble in water. All soluble in water. It means they dissolve in water nicely. Examples? Lila? Is Lila there?
Yes, sir.
What are the examples?
Sucrose.
Karen, you are back. You're saying you're not doing something. You are back now. I'm recording, don't worry if you missed anything. So sucrose, correct. What else?
Lactose.
Lactose. Maltose. Clear. So what are the main sources? We mainly find them in your table sugar and milk. Milk for lactose. Clear. So NB, there's something that you need to know. Huh? You can write that monosaccharides and disaccharides are known as reducing sugars. Monosaccharides and disaccharides are known as reducing sugars. Except, there's one exception. That's why I was saying, except what? Which one won't be affected by Benedict? So if they are reducing sugar, it means they give you a positive test with Benedict's solution. Except what? There's one exception. What is that? It's non-reducing. What is it? So I said monosaccharides and disaccharides are reducing sugars, which means they give a positive test with Benedict's reagent, except, what's the exception, Lila? Which one here is a non-reducing sugar? Except sucrose is non-reducing. You need to know that, huh? Sucrose is non-reducing. So it means it won't work with the Benedict solution. Clear? Benedict reagent. Now let's look at polysaccharides. Poly means many, huh? Polysaccharides. So it's many monosaccharides joined together. Clear? Many saccharides joined together. Many monosaccharides, if you want, you need to put it that way. Joined together to form a complex molecule. To form a complex molecule. To form a complex molecule. Are we together? To form a complex molecule. So it's a long chain, huh? Long chain of monosaccharides. Clear? A long chain of monosaccharides. They are insoluble in water now. Insoluble in water, which makes them, now you can, so for storage, for example, of carbohydrates, or for storage of carbohydrates in plants and in animals, they have to be stored in this format, because it's insoluble in water. Clear? Insoluble in water. What are the examples? These ones are easy. Examples?
We have starch, say.
Starch. It's insoluble, yes. What else? Cellulose, huh? For your cell walls. Cellulose, glycogen. Huh? Remember, talk of in the liver, glycogen. Clear. So sources, mainly bread, potatoes, pasta. Cellulose in plant and animal cells. Huh? Sources, bread, potatoes, pasta, cellulose in cell walls. Clear? Now let's talk of quickly the lipids or fats. What are fats made of? Are they different from carbohydrates in terms of elements? They are the same. Huh? They are made of carbon, hydrogen, and oxygen. So fats are made of the same elements as carbohydrates, but fats are more complex than carbohydrates. Clear? Fats are more complex than carbohydrates. One fat molecule. So one fat molecule is made up of 1 unit of glycerol, not double L, 1L, and 3 units of fatty acids. Clear? 1 unit of glycerol and 3 units of fatty acid. Now, what we have 2 types of fats. I'm sure you know that. We have saturated and unsaturated fats. Huh? What's the difference? Lila, what's the difference? Saturated and unsaturated fats.
Unsaturated fat is liquid at room temperature.
Okay, hold on. Hold on. You said unsaturated liquid at room temperature. Okay, correct. Okay. Yes, unsaturated is liquid at room temperature. You're cooking oil. Okay. Yes, I need to communicate with you. Hold on, guys. Just hold on. Hold on there. Hi, guys. Hi. Can you hear me?
Yes, sir.
So we said
Yes.
this one we're talking of
Yes, sir.
Yes, sir.
liquids, liquids at room temperature. So then these ones are solids at room temperature. Yeah. So these ones sometimes they are not easily digested. They can form a challenge. Okay, the saturated one, the saturated fats. Now, in terms of fats, what are the uses? Fats uses are more the same with carbohydrates, but fats is mainly for energy backup. I'm sure you know that. Uses of fats. So what can we say quickly on fats?
Warmth.
Uses of fats. Fats release, so we can say that fats release large amounts of energy than carbohydrates. They release large amounts of energy than carbohydrates. They are used to make cell membranes. To make cell membranes. Then what else? They are stored under the skin to insulate against heat, so homeostasis. So they are stored under the skin to insulate heat when it's cold. Clear? Are we together? Fats also form a layer around the organs in the body to what? To protect the body from damage. Form a layer around organs to protect them from damage. Clear? Make sense? Now let's look at
Yes, sir.
proteins. Quickly.
Yes, sir.
What can you say about proteins? Proteins, what can you say? What are they made of? Let's start from there. Proteins, what are they made of?
Carbon, hydrogen, oxygen, but they also contain
carbon,
nitrogen.
hydrogen, oxygen, then what?
Nitrogen.
Nitrogen. Stop there. Okay, out together. So you can write that all proteins contain this. Huh? I know where you wanted to say. All proteins contain this. Carbon, hydrogen, oxygen, nitrogen. Okay. But some proteins contain sulfur, which is S, sulfur, and what? And phosphorus. But all of them, they contain what? They contain that. Huh? And this was the smallest unit for a protein?
Acid.
Yes. Smallest unit is amino acid. Clear. Smallest unit is amino acid. So now we've got different proteins because it depends with how the amino acids are grouped or arranged in terms of their bases. Huh? Clear now. Now, what are the uses of proteins? Which you know very well. Uses of proteins. What did you say? Growth. Maybe. Growth. How exactly, huh? So, muscle growth.
Making new body cells.
Okay, I would say, okay, making new body cells. What else? The repairing what? What else? Protein. Uses of proteins. You said making body cells, repairing. When they're making new body cells, that's what you're talking about, growth. Huh? So when they grow, they actually is new cells developing as you grow. Now repairing body tissues, body cells. What else? Proteins. Uses. Oh, didn't you say what is? Can someone define what an enzyme is? Formation of what? Enzymes. What is an enzyme? A biological catalyst that speeds up a chemical reaction that is protein in what? In nature. So making up manufacture of what? Enzymes. Clear? Formation of hormones. What else?
Making antibodies.
Yes. Making antibodies. Exactly. What else? Protein. Protein. Okay. You guys get there? Okay. Well, those are the major ones. Now, let's look at. Now we also have vitamin from your syllabus. I think you're simply required to know A, B, C, not in detail, and D. Organic Okay, vitamins, you know that they are required in small amounts in your human body, but they are very essential, especially vitamin C. Clear, for building your immune system and things like that.
Yes.
Now, vitamin D, where do you get that from? Where do you get vitamin D from? From the sun. We get vitamin D from the sun. We can get it also from liver and from fish. And what are the main functions? How does it help the body? Or what's the use of vitamin D? It helps in formation of what? It helps in bone formation. Clear. Combined with calcium, it helps with iron, it helps in formation of bones. Now, my question, next question, before we look at what we want to look at next. What is a balanced diet? What's the definition of a balanced diet? And we're going to look at food tests. What's a balanced diet?
Balanced diet.
Yes.
Yes. Balanced diet is when it's the consumption of all nutrients in equal amount in the right time.
I said the equal amounts, a diet that contains the correct. You said the correct one?
Correct amount of each nutrient.
Yeah, okay, that's somehow. If it's 3 marks, you won't get. Anyone else? This is okay, but anyone else? A different definition, balanced diet?
A balanced diet is it's a diet consisting of a variety of different types of food, and it provides the adequate amount of the nutrients that are necessary for the health of the body.
For the health of the body. Okay, that's true. 2 marks maybe. A balanced diet is a diet that contains the required nutrition. A diet that contains the required nutrition in their correct proportion. In their correct proportion. So required nutrition in their correct proportions, which include, now when you're doing extended, you have to mention, which include, what are they? You have to mention the 7 of them in your definition, which include. So I said a balanced diet is a diet which contains the required nutrition in their correct proportions, which include, what are they? 7. Which includes carbohydrates, number 1. Number 2, fats. Number 3, proteins. 4, vitamins. 5, mineral ions. 6, water. 7, roughage. Clear? Not difficult, huh? Let's look at food tests. Then we're going to look at digestion before we look at the heart and everything else. Now, food tests. We have to know how to test for what we're talking about. Now, food tests, let's talk of the starch test. Starch test. So remember, I'm talking of food test. Huh? I'm not talking of leaf starch test. That's a different thing altogether. We need to be able to know the difference between. Okay, let's just do it now. Before, so if we do a leaf starch test here, let's do them together here. And here we talk of a food starch test. Now, if it's a leaf starch test, what do I do quickly? What's the first thing? Leaf starch test. So you can tell me what's the first thing we do in a lab? Lila?
Use
Tandy, you have been quiet. Yes? What do you do in a lab? You want to test a leaf for starch. What's the first thing that you do? You boil what? You boil the leaf. In a test tube with water. What's the reasons for boiling? You can tell me what's the reason for boiling. Why do we boil? Yes? I think you're boiling there. I mean, I'm not sure what's happening with your sound. You're trying to communicate. Why do we boil the leaf, Tandy? In the water, it's not to remove chlorophyll either. It's to what? Is to denature enzymes. To denature, maybe your books don't mention that. Is to denature enzymes so that we stop respiration from taking place. Why? Because we are testing a leaf for starch. We don't want the starch to be converted to glucose by respiration. Clear? To be used up by respiration. To denature the enzymes so that we stop respiration from taking place. Clear? That's why we do that. Are we together? Make sense?
Yes, sir.
Because we want, if there is any starch, we want the starch to remain. Because we want to test for starch. So we have to stop respiration because respiration uses up the starch. So we are boiling. Remember, respiration works with enzymes. So we denature, we destroy the enzymes by high temperature. Number two, what do we do? So what's the second thing? Once we have boiled the leaf in water, what do we do? We now boil the leaf in what?
Alcohol.
Yes, we boil the leaf in alcohol. What's the reason now? That's what Lila had said. To remove all the what? The green part is what? Chlorophyll. Because it will affect your starch test to remove all your chlorophyll. Because chlorophyll affects your color changes when because you want to do a starch test. Are we together? So the reason. Now, number three, what do we do? We now boiled that. We now take the leaf. What do we do? What do we do? What's the next thing? Do we now do a starch test? The third thing, what do you do? There's something you do first. Who remembers? Number three, what do you do?
Remove the leaf from the alcohol and dip it into hot water.
That's correct. So you remove the leaf from alcohol and place it in hot water. What's the reason?
To soften it.
Soften it.
To soften it, because it will be brittle, it will easily break. So to soften it. If you don't do that, your leaf will just break and it will be difficult to altogether. It will be very hard and brittle. So to soften it. That's why you just do it for a short time to soften it. And the last thing, you place the leaf on a what? On a white towel. And you add iodine solution. Add iodine solution. If starch is present, it turns blue-black. Clear? Everyone knows that. If starch is not present, it remains brown. Clear? Clear like water. Anyone with questions? Now let's talk of food tests now. How do I test food? For example, I have bread in my house. How do I test it for starch? Different now. So how do you test food for starch? If I give you a bean seed, how do you test it for starch? It's different from if I give you maybe a bread or something. How do you test for a bean seed? You have to crush it first. Huh? Makes sense? Anyway, but how do I test? So I take a food sample, I place in a what? In a test tube. Huh?
Test tube.
So you place a food sample. Oh, sorry, no, it's not even necessary. If you simply you simply place the food sample on a white tile. Clear?
Yes.
Place food sample on white tile. And add what? Very easy. Huh? Add iodine solution. Clear? Then
Yes,
the rest is the same. If starch is present, turns blue-black. If starch is absent, it remains brown. Clear? Everyone? Now let's talk of testing for protein. What do we use? In fact, let's start with the reducing sugars. What we have just learned. Reducing sugars. Reducing sugars, we use what is known as what? Benedict solution.
Benedict solution.
Or Benedict reagent. Okay, Benedict solution. So what do we do? Who knows what do we do? What should we do? Benedict solution. What do we do? Who knows? You place a food sample in what?
Test tube.
A test tube.
In a test tube, maybe. Yeah, that's correct. Place a food sample in a test tube, mix it with water. You mix it with water. Then you put the test tube in a water. Next, number 3, place the test tube in a water bath. Clear. Then you heat, so it's indirect heating. Then you heat the water bath. Huh? So you just want it to be warmer until the solution is warm. Clear. You heat the water bath until the solution is warm. Then you add Benedict solution. Is anyone lost on what you have said? Number 1, Yasha, we take a food sample, we place in a test tube, we add water, we mix together. Number 3, we take that test tube, we place in a water bath. Maybe if I draw a diagram, it will make sense. Water bath like this, with water, then you go to a test tube. That's what we're talking about here. Clear. Then you heat. That's why I said indirect heat. Clear. So we heat the water bath until that test tube, with the solution in the test tube is warm. Clear. Then we add Benedict solution. What's the color of Benedict solution? You need to know it. What's the color? Who knows what's the color of Benedict?
Brown. Brown.
Blue. Not brown. Blue. Clear. Benedict solution, same as Biret, is blue. So we add Benedict solution, which is blue. So now, if sugar is present, what happens? If reducing sugars are present, it turns from what? From blue to what color?
Red or orange.
Before red orange, what happens? They are color changes. This one, that's why you have to be careful. We're explaining an example. From blue to what?
Yellow.
Green.
From blue to green, then to yellow. We need to know these color changes. Huh? Then to orange, then to red. Are we together? Those are the color changes that you should expect, from blue to green to yellow to orange to red. Depending on how we together then. Make sense? Oh, it doesn't make sense. Okay. So that's your testing for reducing sugar that we talked about. You now know what are the reducing monosaccharides and disaccharides, except sucrose. Now, let's talk of testing for protein. What do you use? Burette tests. It's known as burette. So what do? What do? You take a food sample, you place in a test tube. Huh? Place a food sample, place in a test tube. You can even add little water. In some books they say add little water and shake it. Then simply add burette solution, which is blue. Huh? So what are the color changes if protein is present? So you simply shake. So you add burette solution, then you shake it. This one you don't heat. Huh? That's the only one you don't heat. Huh? You simply shake. So now if you shake, what are the color changes from blue to what?
Purple.
Yes.
Purple.
Blue to purple. Blue to purple. Some say violet. Some say lilac. Clear.
Yes.
Now, how do you test for fats? Which is known as the emulsion. What's the spelling now? I'm not forgetting, but anyway, it's known as the emulsion test. I'll check the spelling here. The emulsion test. Now, how do you test for this? It's easy. It's one of the
You
easy.
spelled it correctly.
You place a food sample in what? In a test tube. Is that what you wanted to say? Place a food sample in a test tube. You can even mix with little water. Then you add what? What do you add? Ethanol, huh? Which is colorless. We add ethanol, which is colorless, and we shake it. Huh?
Mm.
If fats are present, this turns what color?
Cloudy white.
Cloudy.
Yes, milky or cloudy white. If fats are not present, it remains what? It remains colorless. Clear?
Yes.
Okay. So that's all you need to know. Now, I know we've got 7 minutes. I need to look at the first part of this. I'll give you some.
So,
Yes? Question?
in our syllabus, do we need to know how to test for vitamin C?
Partly. Not the details. Okay, we'll talk about yes in your syllabus. I'll just I think I saw yes. Yes, actually. So yes, you reminded me there. Okay, we'll look at that. So you have reminded me. Anyway, we'll look at that. I'll show you. It's not difficult. Now, digestion. I think that's. So we'll look at that the next. Now, digestion. So, what is the difference between chemical digestion, quickly, and chemical and physical digestion or mechanical? What's the difference between chemical and mechanical digestion? Mechanical, it means physical, and chemical, what's the difference? If a question says compare and contrast, I haven't seen a question, but perhaps they can ask you anything. Compare and contrast chemical and mechanical. Karen and I, there in the lesson, you've been very quiet. Oh yeah, absorbed.
Yes, sir.
What's the difference between chemical that's physical? I mean chemical and physical. What's the difference?
Mechanical digestion involves physical movement to make foods, like break down the foods into smaller pieces, and chemical uses enzymes to break down the food.
Okay, so use of enzymes. I simply wanted simple answers. Use of enzymes, no enzymes involved. Huh? Involved. Now with this one here, new product is formed. Huh? New products are formed. Clear? With this one here, you are simply breaking down no new product. Huh? No new product. You are simply breaking down in same product into smaller clear. Make sense? Now, let's talk of now. So when we do this, we need to know the elementary now. I'm sure you know that. To be able to label it and to be able to know. So in the mouth, what happens? Let's start there. The mouth, you can tell me what happens. What's the first thing that happens? Physical digestion. Huh? Remember your teeth, you break down food molecules into smaller. So the first thing that happens in the mouth is physical digestion action by your teeth to break down food molecules into smaller molecules so that it's easy for enzyme digestion. You're actually increasing, when you're chewing, you're increasing what? So mechanical, we said mechanical digestion action of the teeth. Teeth, what does it do? It breaks down what? It breaks down large food molecules into small molecules, increasing surface area for enzyme digestion. Clear? So it actually helps. Clear? Everyone there?
Yes.
Now, then what happens after that? The salivary amylase, what does it do? Salivary gland, sorry. It secretes what? It secretes salivary amylase. I'm sure you know that. Huh? What does salivary amylase do to this food?
So put down the
What does salivary amylase act on protein? Does it act on fat? No. Huh? So what does it digest from this food that is in the mouth? It digests what? Starch. It breaks down starch. So starch plus salivary amylase, the enzyme. It breaks down starch to what? Who knows? Breaks down starch to Tandy. Salivary amylase breaks down starch to
Mouth.
maltose. Maltose. TOS. Maltose. Then after that, food goes down the what? What does food go down?
Esophagus.
Oh, esophagus.
Esophagus. What happens there? In the esophagus? How does food move?
Peristalsis.
By peristalsis.
Peristalsis.
What's peristalsis?
Muscular. Like
The
weight
contraction,
motions.
yes, contraction and relaxation of antagonistic muscles. Muscles of clear to move food down. Huh? So they always do the opposite. If the longitudinal is relaxing, the other one is contracting until the food moves down to the what? To the stomach. Clear? Now, the last thing before the next lesson, in the stomach, what happens? You're saying the stomach produces? Okay, the stomach walls, okay, that's correct. Stomach walls secrete hydrochloric acid. Yeah? Hydrochloric acid. What are the uses of hydrochloric acid? Okay. Of hydrochloric acid. What's the usage quickly?
It makes the stomach alkaline condition.
Acid. It makes the stomach, correct your statement, acidic condition. Huh? It provides,
Acidic
yes, it
condition.
provides the acidic condition. Why do we need acidic condition? Acidic condition to prepare the stomach for digestion of protein by protein. Clear? Then it also kills what? What does it kill? An acid?
Bacteria.
Yes, it kills bacteria. From food from the mouth, kills bacteria. Clear? Then the stomach was also secrete, second point, the stomach was also secrete protease, which is the enzyme that will break down protein. So protein, that's the question here, plus protease, we form polypeptide, not amino acid, polypeptide. Clear? Polypeptide. Anyway, we have a recording of this whole thing. If you missed anything, it's a very important lesson. We build it from there. Clear?
Yes, sir.
To know what happens until we reach the end point. Now we are done. See you for the next lesson. What's the next lesson, by the way? What's the next lesson? Can someone remind me? Huh? Comments. Followed by what? Extended method. Okay.
Yes, sir.
Okay.
Thank
Yeah, okay.
Thank you, sir.